Now mitochondria. This is the structure of mitochondria. Mitochondria is made up of two membranes, outer membrane and inner membrane. This is outer membrane, which is smooth, and inner membrane is folded. This inner membrane, these are fold. Inner membrane is folded, these are folds. So outer membrane, inner membrane, which is folded, and these folds are called cristae. And the cavity enclosed by two membranes, this inner cavity, this is called matrix or mitosol. Mitochondria is the site of cellular respiration and ATP synthesis. Cellular respiration occur in mitochondria, and ATP synthesis occur in mitochondria. For this reason, mitochondria are considered as powerhouses of cell. Electron transport chain and oxidative phosphorylation, these processes occur in inner membrane, inner mitochondrial membrane. Electron transport chain and oxidative phosphorylation, these occur in inner mitochondrial membrane, and this inner mitochondrial membrane is folded because to increase the surface area. If these folds were not present, then there will be small area, and enzymes of electron transport chain and oxidative phosphorylation, the enzymes of these processes are present in inner membrane, and there is more requirement of in these enzymes. So this inner membrane is folded so that there should be more area where more enzymes should be present, so that more and more these processes, oxidative phosphorylation and electron transport chain, these processes can occur at a high rate. ही जेका इनर मेम्ब्रेन आहे हिन में ही फोल्ड आहिनि, ही फोल्ड हुन करे आहिनि त जीअं हिन जी सरफेस एरिया वधे, छाजे करे जो इन इनर मेम्ब्रेन में इंजाइम्स आहिनि न इलेक्ट्रॉन ट्रांसपोर्ट चेन ऐं ऑक्सीडेटिव फॉस्फोराइलेशन जूं, त ही फोल्ड आहिनि त जीअं सरफेस एरिया वधे त जीअं उन्हनि ते वधीक इंजाइम अची सघनि, जेतिरियूं इंजाइम वधीक हुजनि त जीअं ही प्रोसेस इलेक्ट्रॉन ट्रांसपोर्ट चेन ऐं ऑक्सीडेटिव फॉस्फोराइलेशन इहे वधीक इफेक्टिवली इफिशिएंटली थी सघनि. And enzymes for metabolism of carbohydrate, lipids and proteins. Carbohydrate, lipid and protein the metabolism जो enzyme, these are present in matrix. Means in other words, citric acid cycle occur in matrix. And enzymes for heme synthesis and urea synthesis are also present in matrix. Matrix contain DNA, mitochondrial DNA. Mitochondria contain its own DNA, that's called mitochondrial DNA, which is present in matrix. In matrix there is DNA is present, RNA is present, and ribosomes are also present. In other words, the whole machinery of the protein synthesis जो जेको भी मशीनरी आहे यानी DNA, RNA ऐं राइबोसोम्स, इहे माइटोकॉन्ड्रिया खे पंहिंजा आहिनि, उन करे माइटोकॉन्ड्रिया प्रोटीन ठाहींदो आहे. ऐं about 10% of mitochondrial protein is made up of mitochondria itself. 10% mitochondria जी प्रोटीन माइटोकॉन्ड्रिया पाण ई ठाहींदो आहे. Now endoplasmic reticulum. Endoplasmic reticulum is network of membrane enclosed spaces which is present or scattered throughout cytoplasm. तव्हांखे सम्झ में आयो? Membrane enclosed spaces. This is one membrane enclosed space. This is space, this is membrane. A membrane, a space enclosed by membrane. This is another membrane enclosed space. This is another membrane enclosed space. In this way, endoplasmic reticulum is network of membrane enclosed spaces. These membrane enclosed spaces form a network which is present throughout cytoplasm from nucleus to cell membrane. This is suppose nucleus, this is cell membrane, this endoplasmic reticulum, this is present in this fashion. scattered. Endoplasmic reticulum is scattered throughout cytoplasm. Like this. This is endoplasmic reticulum. This is from nuclear membrane up to cell membrane. This is scattered throughout cytoplasm. There are two types of endoplasmic reticulum: rough endoplasmic reticulum and smooth endoplasmic reticulum. On rough endoplasmic reticulum, ribosomes are present. They give a rough appearance to the endoplasmic reticulum. That's why this is called rough. And rough endoplasmic reticulum synthesize protein. And functions of smooth endoplasmic reticulum are synthesis of lipid, detoxification of drugs, and storage of calcium. Now, Golgi apparatus. Golgi apparatus is cluster of vesicles. Cluster of vesicles. Suppose this is a vesicle. This is another vesicle. This is another vesicle. Such vesicles form a cluster, and this cluster of vesicles is called Golgi apparatus. And each vesicle is called dictyosome. These vesicles are called dictyosomes. When protein is synthesized in rough endoplasmic reticulum, newly synthesized protein from rough endoplasmic reticulum is forwarded to the Golgi apparatus. In this fashion, the newly formed protein in rough endoplasmic reticulum is forwarded to the Golgi apparatus, and Golgi apparatus add carbohydrate and lipid to that protein, and this modification is necessary for transport of that proteins across the cell membrane. Newly synthesized protein, which is synthesized in rough endoplasmic reticulum, that is sent to Golgi apparatus. And what Golgi apparatus do to the newly found protein? Golgi apparatus add some carbohydrate and lipid to the protein, and this modification is necessary so that this protein can move across the cell membrane easily. Lysosomes and peroxisomes are formed by Golgi apparatus. See here carefully. Suppose this is Golgi apparatus. How lysosomes and peroxisomes are formed from Golgi apparatus? In this fashion. These vesicles this, and further this separates, and this become like this, and this vesicle separates, and this forms lysosome or peroxisome. Lysosomes and peroxisomes are formed in this fashion from Golgi apparatus.